Hello, everyone. Welcome to the new era of medicine, mRNA vaccines. Let's take a step into the future with this technology. I'm Alison Simpkins, and I worked on this project along with Sarabeth, Hoffman, and Irene Labarta. Our project author and advisor is Dr. Scott Diamond. The story begins in 1978 with a scientist in Hungary who had the revolutionary idea of using mRNA as a potential new vaccine technology. This scientist, Dr. Katalin Kariko, joined the faculty at the University of Pennsylvania and worked alongside Dr. Drew Weissman during the 1990s and early 2000s. Much of this work is published and patented, and it has made mRNA vaccines a revolutionary and effective new vaccine technology. This story jumped to the forefront of science with the emergence of the novel coronavirus SARS-CoV-2 and its associated disease, COVID-19. The solution to this pandemic is vaccination. A little bit of background on mRNA vaccines and how they work. A protein associated with the virus is isolated. For the case of this, we use the full-length SARS-CoV-2 spike protein. The mRNA encoding this protein is made, and it is encapsulated in lipid nanoparticles, or LNPs, which is the delivery system for the vaccine. Following injection, an immune response is then generated. This immune response prevents against future infection, as well as severe illness in the case that infection still occurs. mRNA vaccines present the most efficient way to meet the current demand. They are faster, safer, and cheaper to produce than other types of vaccines, as they use a cell-free synthesis of mRNA, and the process is independent of the encoded antigen. There is a global need for a scalable and agile mRNA vaccine manufacturing process to meet the large demand to end the current global pandemic and treat other diseases. Currently, there is no published information or shared intellectual property on a manufacturing process to produce an mRNA vaccine.
Thank you, Alison. Our goal was to manufacture 200 million sterile doses of the COVID mRNA vaccine in 13 weeks, and to publish a report on this. In order to do this, we firstly studied each part of the mRNA production process. We determined which techniques to use by evaluating laboratory scale literature and patents, and we assembled this into a manufacturing process. Every week, our process produces 15.4 million doses. We input 11 grams of plasmid DNA, and we use over 100 manufacturing steps, utilize over 30 different raw materials, and use over 300 streams to produce the 15 million doses on a weekly basis. Our manufacturing process is broken down into 5 steps. Firstly, the production of the plasmid DNA that has the gene of interest, which is outsourced. Then the mRNA synthesis process, followed by mRNA purification, the encapsulation of mRNA into the lipid nanoparticle, and lastly, packaging. This whole process is supported by quality control steps. On this slide, you can see our total process flowsheet diagram with its over 100 steps. Firstly, the plasmid DNA is outsourced and stored into a freezer. This is fed into a wave bioreactor to produce the mRNA strand that is then purified through a series of steps. The mRNA is fed to a microfluidics device from which it is encapsulated into a lipid nanoparticle that is then also purified through a series of steps. Lastly, this product is frozen, filed, labeled, packaged, and shipped in thermal containers to vaccination sites. The 2 main parts which make our process novel and innovative are the use of a cellulose chromatography column. We've scaled this technology from the laboratory scale of 780 milliliters into our process, which has a unit volume of 1,000 liters. Secondly, we use the physics of microfluidics devices to provide a mixing chamber for the lipids and the mRNA using a novel patent. I will now pass it on to Sarabeth.
Thanks, Irene. So our process contains 6 major unit ops. In order to find the price of each equipment, we had to contact vendors to obtain a quote. So first, we're going to start off by talking about our in vitro transcription reactor, which is the first unit op in our entire process. The purpose of the IVT reactor is to produce mRNA from a DNA template via the enzyme RNA polymerase. We determined the in vitro transcription reaction will take place in a wave bioreactor system, which consists of a rocker upon which a disposable plastic bioreactor bag is placed. For this process, per batch, we determined the reaction volume is 110 liters. So following the in vitro transcription reactor, our next major unit op is our tangential flow filtration system 1. The purpose of this unit op is to remove all elements from the IVT reaction. Water is used as a buffer to purify and prepare the material for the chromatography column. In addition, the membrane has a molecular weight cutoff of 100 kilodaltons, and this is to prevent the mRNA from passing through the pores of the membrane. And lastly, we calculate the size of the membrane to be 6.3 meters squared. On the next slide, we come to our next major unit op in our process, which is our cellulose chromatography column. The purpose of this unit op is to remove double-stranded RNA, which is a byproduct of the IVT reaction, and this is done by binding the double-stranded RNA to a cellulose resin in a chromatography column. We calculated the reaction volume of the column per batch to be 985 liters. Following the chromatography column, our next major unit op is our tangential flow filtration system 2. The purpose of this operation is to perform a buffer exchange with sodium acetate. The reason sodium acetate was chosen to be the buffer of TFF2 is because this will prepare the RNA solution for the LNP formulation process that occurs in the microfluidics devices downstream. In addition, the size of the membrane is 6.1 meters squared. So we come to our next major unit op after TFF2, which is our microfluidics mixing devices. The purpose of these mixing devices is to form the lipid nanoparticles and encapsulate the mRNA in the LNPs. Staggered herringbone microfluidics mixing devices will be used to produce the mRNA containing LNPs. One inlet stream is the mRNA in the acidic sodium acetate buffer, and the other inlet stream is lipids dissolved in ethanol. The two inlet streams will meet at a mixing channel where mixing and LNP formulation occurs on the order of milliseconds. After mixing, there'll be a single exit stream which contains the LNPs encapsulating the mRNA product. To process 5,000 liters of volume per batch, the setup of these microfluidic chip devices consists of 25 sets of 16 parallelized chips. The last major unit op in our process is the tangential flow filtration system 3. The purpose of this operation is to perform a buffer exchange with PBS to incorporate this solution into the nanoparticle since it contains components needed in the final pharmaceutical formulation. In addition, the size of the membrane was calculated to be 87 meters squared. So finally, we come to our quality control steps, which are needed to meet FDA approval. So first we have our RNA gel electrophoresis, and this is to check that approximately 90% of the RNA is full length. Next, we have our dynamic light scattering, which is to check that the LNPs are approximately 80 nanometers in size. Finally, we have our LAL assay to check that the endotoxin levels in our product are less than 10 endotoxin units. Next, Alison will talk about our financials.
Thanks, Sarabeth. The cost to produce a single dose of our mRNA vaccine is $1.87. After analyzing Pfizer's current pricing strategy in the US market, we settled on a price per dose of $15. With our current manufacturing process, we're able to produce 725 million doses per year, which results in a net sales of $9.79 billion per year. The resulting high profitability gives us the ability to underprice the vaccine when selling in other markets, thus making the vaccine affordable for nations unable to pay the US selling price. It also allows us to be highly profitable at selling prices as low as $6 per dose, as seen in the graph on the right. This is a highly attractive venture, which requires a total capital investment of $746 million. Additionally, we have net earnings of $4.47 billion per year, a return on investment of 475%, as well as a net present value in 2021 of $19.6 billion. 30 years after Dr. Carrico performed her revolutionary research at UPenn, the story of mRNA vaccines continues. We are just at the start of a new era of medicine. With our proposed manufacturing process, we can produce 200 million sterile doses in 13 weeks, as well as 725 million doses on an annual basis. This meets the production objective set by the United States government and Pfizer. Additionally, this constitutes a highly attractive business venture. And third, our report, which will be published by Penn, outlines this manufacturing process, allowing companies other than Pfizer and Moderna to establish these types of manufacturing capabilities. Finally, the proposed COVID-19 mRNA vaccine manufacturing process we present here today, in fact, outlines the manufacturing process for the production of any RNA vaccine, as the only main thing that changes is the mRNA sequence. This can be easily used to produce vaccines for current SARS-CoV-2 mutants, future COVID-19 booster vaccines, and vaccines for other diseases. We truly hope that UPenn continues to set the pace of this new era of medicine. Thank you so much for joining us and listening to our presentation. We greatly appreciate it.